First, I want to introduce some people in my lab who did the work. Nick Hudd, he's one of my best postdocs. Nick is my close collaborator. And a lot of what I'm doing has to do with our interaction. But I'm going to talk about work from Anton Petrov, second right there, Nick Kovacs, Catherine Lanier, Jessica Bowman, and then my collaborators. I have some very nice collaborators at Georgia Tech, Nick Hudd, Roger Wortel, and then George Fox, who's at Houston. And I'd like to thank the organizers, Irina, wherever you are, for inviting me here and running such a great meeting and keeping it so organized. Of course, this is Japan, so of course it's organized. And I'd like to say hello to my friends in California at the NEI. Good afternoon. So, oh, that's going to be annoying. What we do is we try to understand the origin of life from extant biology. And we look at the tree of life, or the circle of life, I guess, at this thing, and we look all around this thing, and we try to use that to walk backwards in time. And there's a lot of people who do this kind of thing. But we, I think, probably go back further. And one of the ways we can do that is that we focus on structure, on three-dimensional structure. There is this axiom, I guess you could say, that structure is more conserved than sequence, and if you want to look far, far back in time, you use structure instead of sequence. So you can have, especially with nucleic acids, you can have RNAs in which the sequence is essentially scrambled between two RNAs, and yet the structures are identical, essentially, within the experiment. So you can have variation in sequence, but structure is conserved. If you want to look far back in time, you use structure. Okay, let me see if I can make this work. Okay, so this is basically what we're after. This is the tree of life. We're interested in LUCA, and what was LUCA? We're sort of interested in that, and then where did LUCA come from? Basically, this is the process, and we're focused on the translation system. And sort of for background, you should know that the translation system was done at LUCA. So when you talk about the origin and evolution of the translation system, you're really talking about things that happened before LUCA. So if we can understand the origin of the translation system, we are looking really far back in time, 3.8 or 4 billion years ago, depending on when life originated. So of course, this is what everybody does when they look at the origin of life, and this is sort of various models people have. There's RNA world, clay world, metabolism world, vents, and membranes, which we've heard about some of this at this meeting. And I probably haven't really done justice to these cartoon representations, but these are probably not all the models, but this is kind of a survey of the model. And then we have the data, and this is the data that we focus on, which is what I call the universal gene set, things that everything alive has. And it's actually, there's several amazing characteristics of this universal gene set. And this is from Doolittle's paper in 2004, but Pace has looked at this, and Kunin, and there is a nice consensus pretty much on what the universal gene set is. And these are basically genes that you can find orthologs in everything alive. So this is what you would say is universal biology. Everything has this. And the first thing about it is it's a very small list. It doesn't have very many genes on it. It depends on whose version you have, but it has between 30 to 50 genes. It's a small list, and I've color-coded them here by what they're involved in. And the pink is translation, and then we have replication, transcription, and replication. And basically, the point of this thing is that this is dominated by translation. So the universal gene set of biology is dominated by translation. And this list actually does not have genes that encode RNAs. So there are no genes for tRNAs on here and ribosomal RNAs and things like that. If you actually included those on this list, it would be a longer list, and there would be more pink. Okay, so the universal gene set of life is dominated by translation. That's the point of this. And so you have this problem here that you have the data that sort of tells us about LUCA, and then you have these models, and the models don't fit the data in the sense that let's say RNA world, for example. The RNA world predicts ribozymes that can replicate themselves and do metabolism and things like this, and there's nothing like that here. There's no integral membrane proteins. There's no proteins involved in membrane biosynthesis. These models are totally disconnected from this data. And so what should you do? You have a model and you have data and they don't fit. Generally, people would throw away the data. That seems to be sort of what has happened in the origin of life. People who study the origin of life basically ignore the universal gene set because it doesn't seem to relate to their models. So when we look at this, basically, our idea is I just ignore all the models. I just want to use the data, and I want to walk the data back and say, what does the data tell me about the origin of life? I don't care about RNA world. I don't care about metabolism world. I just want to know what this means. Number one, I want to know, why is it so small? Why is it dominated by translation? And can we use this to walk back? And an important thing about this list is these are sequences, right? These are sequences that encode proteins, but we know the structures of these proteins, right? Crystallographers have determined ribosomal structures. We know this, so this isn't just the gene set of life. This is really the sort of structure of life. We know the universal three-dimensional structure of biology, the conserved part. Another thing I'll point out about this list, I'll sort of poke Eric a little bit. There, okay. People focus on things that are universal in biology, but because there is this disconnect between the data and the models, they tend to focus on small molecules. So you'll hear people talk about metabolites and small molecules of biology that are highly conserved, and they'll ascribe importance to these things, but they never look at the macromolecules, which are universal biology. And the reason I think people don't do that is because this doesn't fit the models. And it's very hard to take these universal macromolecules and fit them to these models. There's just no relationship between them. Okay, so this is what we do. We're looking at universal three-dimensional structures. And we do that, as I said, because structure is more conserved than sequence. If we want to look far back in time, we use three-dimensional structures. And we're asking, what is the universal three-dimensional structure of life? How conserved is it? Sequence is kind of digital, essentially. You can say this is a C, this is a G. But when you're talking about structure, you're talking about something that is continuous. You have RMSDs of atoms or something, and it's a little bit, it's actually significantly more difficult to sort of be analytical and quantitative about structural conservation than it is about sequence conservation. So we have to sort of work and figure out how to deal with that. We want to know how structure is elaborated. Like we have things that are conserved, but then the part that varies. What are the rules of that? And then what are the origins of these conserved structures? That's really what we're after. So what I'm showing you here are secondary structures of ribosomal RNAs. First thing, these are very large molecules. They're huge. And this is E. coli, yeast, fruit fly, and humans. And I want to first focus on, let's just look at that little purple thing right there, that little knob. And what I want you to notice is E. coli has it, yeast has it, fruit fly has it, Homo sapiens have it. Everything in biology has that. Okay? And we have sequences of many ribosomal, the data is very rich here. And we have three-dimensional structures. And we can say that everything in biology has that little knob right there. And, but what really we want to look at is the three-dimensional structures. So what I want to do is I want to show you kind of what we mean when we say the universal structure of biology. So I'm just going to take a part of this thing. We have crystal structures of all these and show you what it means to be conserved. Oh yeah, what is this? Oh, okay, I fixed it. So this is part of the ribosomal RNA from E. coli, and I've stripped out all the bases. It's just a trace of the backbone, and it's beautiful. Okay, if you look at And this is a global superimposition of ribosomes. This is archaea. Okay, so these are two separate branches of the phylogenetic tree. And you can see that these look pretty similar. Now, the interesting thing is the sequences are not necessarily the same, right? Like this is a helix here. And a helix can accommodate many sequences, right? So the sequences are not fully conserved, but the structure is. So these are two branches of the tree of life. I can look at the other one. Here's yeast. And basically, this is it. This is the diversity in all of biology that you're looking at. This is how much this thing changes. And I used to say this thing is how much it changes over 4 billion years of evolution. But Greg Fournier pointed out that I need to double that because the distance from Luca to E. coli is 4 billion years. The distance from Luca to Haloarcula is 4 billion years. So this is 8 billion years of evolution. And this is showing you how much the structure changes. And then just to kind of top it off, you know, evolution sort of is on steroids in organelles. And things are happening much faster in organelles than in cytoplasm. And so here is the mitochondrial ribosome from Homo sapiens. And this, you know, basically this is just cemented. This is, okay, there's some geologists here, so I'm going to say something. You can correct me. This is the most permanent thing that is not cold in the universe. I don't know if that's true, but it sounds good. You can, but it really is very, very constant. Okay, so what we have done is this is just part of the ribosome. We have looked at the entire ribosome, and we've defined what we call the common core. So this is the bacterial, this is E. coli ribosome. This is large subunit RNA and small subunit. And the blue parts are the part that are structurally conserved in everything alive. Okay? And Chad Bernier is a graduate student who actually graduated, but he spent years, to work this out involved superimposition, alignment. It was a sort of iterative process of figuring out what's what. And so, like, for example, this is this little blue bump I told you that everything has. It's right there and right there. But then, like, let's focus on this little arm here. See, E. coli has an arm there, and Haloarcula doesn't. So that's black. So the things that are common are blue. And really, the point of this slide is that, to a first approximation, the E. coli ribosome is the universal core. Everything in biology has something equivalent to the bacterial ribosome, okay? And you can see, it's not exactly true because the black parts are where that breaks down, but to a first approximation, everything alive has a E. coli ribosome inside of it. You do. We all do, okay? All around this tree. That's 8 billion years of evolution. Okay, but now I want to focus on the parts that are different. Because you can look here, like, look at the human ribosome has these devil horns, right? Look at those devil horns. And of course, bacteria don't have that. And so there are places the ribosome varies. And I want to focus on that for a minute because what I want to do is figure out how the ribosome changes over time. And it turns out that the rules are very strict. And there are very specific things that can happen to the ribosome, you know, like nothing can happen there. That's just not allowed, okay? But this helix right here, that's a lot of things are allowed there. In fact, I have, okay, let me just, really what you have is, you have something we call the universal common core, and that's the E. coli ribosome, basically. And then you have a eukaryotic shell, and those are these sorts of things. And then in metazoans, especially in mammals, you have these tentacles that go out. So really there are, I'd say in extant ribosomes, there are these sort of three phases. There's the common core that everything happens. Then there's a eukaryotic shell, the first shell. And then there are these metazoan expansion segments. So this is the large subunit we're looking at. This sort of summarizes the evolution of the ribosome. And we know this because we have examples all over the phylogenetic tree of these things. Okay, now I want to focus on this helix. This is called helix 25 in E. coli, but it's called expansion segment 7 in eukaryotes because It's no longer a helix once you get to eukaryotes. It gets really enormous. In fact, look at human. It has these tentacles. Human has really long tentacles. The ribosome is kind of like kudzu. Actually, kudzu is a Japanese vine that was imported in the United States, and it grows like a foot a day. And the eukaryotic ribosome kind of reminds me of kudzu. These arms are changing very rapidly over evolution, and they seem to be getting longer and longer. But what I want to do is I want to look at these in 3 dimensions. We know how this grows. Everything I'm showing you here is in 2 dimensions, but we know what this looks like in 3 dimensions. And so this is what we call the Vishnu basement. This is helix 25. This is the bottom. This is there everywhere. It never goes away. And that's, we know what that is from bacterial ribosomes. And we have E. coli and Thermus thermophilus. We have crystal structures of quite a few bacterial ribosomes. And then when you look at archaea, you can see helix 25 is still there, and then something has grown out. So this allows us to have an estimate of LUCA, right? Because we say what is common between archaea and bacteria is our best guess for LUCA, and so we say LUCA had helix 25, because that's what's common between archaea and bacteria. That's sort of the standard way you look at things. Okay, so, but we can just keep walking here. Here's archaea. Here's yeast, and what you can see is that the archaean element is essentially in yeast, and then things have grown out of it. Here's fruit fly, and fruit fly looks, I mean, really there's common ancestors here. I don't want to give the idea that fruit fly evolved from yeast, but essentially the common ancestor looks like yeast, and then things have grown out of it. And then it's not nice. We have a lot of crystal structures and cryo-EM structures, and then you can see the same thing. So you guys, this is really important to us when we made this discovery that this is how the ribosome changes, because this is a process called accretion. It's just like geology. It's just the way trees grow, that the ribosome accretes. And when it accretes, like this helix 25, it's always there, right? When you add things onto the ribosome, you don't mess with the underlying structure. That's what evolution does. And we have lots of examples of this over the, I don't want to take time here, but you can just sort of see it. It looks like a movie of things going, okay? So the ribosomal RNA, I need to be careful. The ribosomal RNA grows by accretion. Okay, and when it does that, when it increases in size, it leaves fingerprints. It's very nice. It's like a tree, and when you have a branch growing out of a tree, there's a knot. And so even if you cut the branch off, you can cut into the tree and you could say, yes, there was a branch here once. And so this is something we've identified called an insertion fingerprint, that it's a specific structural element that allows us to say, this green thing grew out of that red thing. And we can do that because we have these structures of, I mean, we know that's true because this is the blue is the prokaryotic ribosome, and this is the eukaryotic ribosome, and we know that the red was essentially, that the blue was the ancestor of the red and green. So, you know, this is not modeling, this is structures, and we really know how the ribosome changes over time. So we have 2 things. We have the ribosome grows by accretion, and when there are growth steps, we don't know that all of them, but many of the growth steps leave fingerprints. So that even if you only had the human ribosome, you could look and say, there was a growth event here. But we don't have to do that because we have many, many ribosomes of all different sizes and shapes. Okay, so now I want to use this tree analogy because the ribosome is very similar to a tree in that a tree records its history, and it does that because it grows by accretion. If the tree died out every year and regrew the next year, then it wouldn't record its history, right? The reason a tree records history is that something happens, and then that doesn't change. You add layers and layers, you add twigs and branches, and you don't perturb the underlying thing when you have growth processes. And so maybe if you're from Arizona and you haven't seen a tree before, this wouldn't make sense to you. But for the rest of us, we can look at this tree, and you could, at a very high level of detail, ascribe relative age to everything on this tree. Right? You could say the oldest part of this tree is in the center of that trunk. The leaves are the most recent. The small twigs are younger than the big twigs, et cetera, right? And you can look at this tree and sort of work out its history. And it turns out, I mean, I've explained a little bit to you, but the ribosome is the same thing, okay? Because it grew by accretion and because it leaves telltale signs when it undergoes a growth event. You can look at the ribosome and you can read out its history just the same way you can read out the history of this tree. So what this really means is there is a history of biology before LUCA, because the ribosome was done at LUCA, and we have ways of reading out the history of that ribosome. So this is, just to be really clear, these are observations. The modern ribosome, it grew, and it is growing by accretion. And we know that because we have crystal structures of so many ribosomes across the phylogenetic tree. So that is what we call an observation. We know that when growth events occur, some of them, not all of them, but some of them leave fingerprints. Okay? So this is what we know. And then these are our inferences. So we are assuming that the common core also grew by accretion. Now, we don't know that because we can't watch it the way we can watch eukaryotic ribosomes, but we're just assuming the process is continuous. And we're also assuming that in the common core, that growth events left fingerprints. So we're just assuming a kind of continuity. If our assumption is wrong, then it's wrong. But I just want to be clear about what our assumptions are. So based on that, we can look at the common core. So this is the thing that everything in biology has, and we can work out how it grew. So I'll just focus on right here. Every time the color changes, there is an insertion fingerprint here. Okay? So you can say something, there was a growth event, that green, then the red got added, that green got added, etc. So we can see these insertion fingerprints, and we're assuming that it grew by accretion, so we can work out in a very high level of detail the steps in the growth of the common core. And so, see this red thing right there? That's actually, the secondary structure is misleading. That's a continuous helix. This is your mother. Okay, that is the beginning of the ribosome. That is the first thing that happened. And then there's a series of growth events that ultimately lead this. But we can work out in a very high level of detail how the ribosome grew. In fact, it's so much detail that it was just mind-boggling, and we just kind of left it for a while. And then we said, okay, let's simplify it. So we grouped these things into phases. We just kind of did a coarse graining of this to cut down on the amount of information. So really, we have these steps 1, 2, 3, 4, 5. We just call that, we just sort of grouped them by color. And it was a little, we could have, so we have 5 phases here. We could have done 4, or we could have done 6. That's arbitrary. But we just had to kind of cut down on the amount of detail because it was just more than we could deal with. So this is sort of a more, so the blue is the first thing that happened. Dark blue, light blue, green, then all those colors. Okay, and we have large subunit and small subunit, and we have ways of correlating. This is published, so I don't want to go into it, but we have ways of correlating between the two subunits. So we have a very, very detailed model for the evolution of the ribosome. And in our lab, what we do is we make these things. I mean, we have an experimental lab. We have predictions on what these molecules should do. We can make any piece of RNA, and we just make them and test the predictions. And so that's really one of the main focuses of my lab. And this is, the nice thing about RNA is that it tends to be modular. And so because we have this structural map of what has happened, we also have a functional map. Okay, because the structure and the function are so easy to decipher. So we have a very detailed map. And all of these, this is a hypothesis. And we can make all of this easily. We can make each one of these molecules. We can test the predictions about what it should be doing. So this is all experimentally grounded. Okay, but I want to change subjects totally, because so far I've talked only about RNA. And we've just talked about the changes of RNA. How much time do I have, Batul? Or who's in charge of the time? I'm fine, okay, because I have 47 more slides. Good. She said I'm fine. I don't really. You guys, so basically everything I've talked about so far is published, but this is new, and this is what I'm really interested in, is the proteins. I haven't talked about proteins yet. We've only talked about RNA. So what this is the three-dimensional structure of the bacterial ribosome, and we have color-coded the RNA by those phases. So the oldest thing here is dark blue, then light blue and green, et cetera, okay? So that the RNA is colored by its relative age. And then the gray things here are the ribosomal proteins. Okay? And what we have done is we have used the RNA to date the proteins. And we just made kind of a simple assumption that, that if a part of protein is surrounded by RNA, then it's of the same age. And this is kind of a test of our model because I'll sort of show you why. But if we can date the ribosomal RNA and we know what we're doing there, then we should be able to date the ribosomal proteins, and we... Okay. All right. Okay. So what I have over here is I'm showing the ribosomal proteins, and I faded out the RNA. And you can see the ribosomal proteins they're unique in all of biology, in they have these... They're not random. I mean, they're not random coil because random coil is in an ensemble. These are in specific states, but they're non-canonical. So they're frozen. They don't move, but they're not folded in normal ways. And you just don't see proteins doing this in biology. And what we've been able to do is we take a given protein, like this one, and we cut it into segments. And we cut it based on the age of the RNA around it. So we basically say this part, in fact, this color is one age, that's another age, that's another age. And we do that by the RNA that is surrounding it. Okay? And that turns out to be pretty easy and clean. There's some places where it's ambiguous, but it's a pretty clean way. And there's a lot of proteins in the ribosome, so this is a lot of data. But this is kind of what it looks like. So this is a ribosomal protein. And this red part is the oldest, and then this yellow part is younger, and that is younger, okay? So we can segment these proteins by age, and so these segments here are all of the same age. These yellow are of a different age, and time is moving in this direction, right? So, and we didn't build this in, we didn't sort of know it was going to be like this, but this is basically a reaction coordinate of protein folding. Okay, what we have here is frozen random coil. I have another? No. Okay, we have tons of these proteins, right? There's a lot of them. We have a lot of data here. And really in this, what we call phase 3, that's the earliest phase where we see protein. The actual core of the ribosome itself has essentially no protein. So when protein starts to show up in phase 3, it's never folded. There's no alpha helices, there's no beta sheet, there's no hydrophobic cores, there's nothing like that. It just looks like that. And then the second thing we have is these isolated secondary structural elements. And they tend to be beta sheets, not alpha helices. And then in the next phase, we see collapse of these secondary elements to globular structures, and then those globular structures elaborate. And the nice thing is, I'm only showing you the prokaryotic ribosome. If we add the eukaryotic, there's that shell. And that shell came after 1.5 billion years. So we have another layer out here where we can say what happened. And you can really see that protein folding. So this is a reaction coordinate for protein folding. Maybe I'll just, okay, jump to this. I'm just going to jump ahead. We have a bunch of statistics, so I'm just going to ignore all that. But this is what it looks like. This is the ribosomal RNA colored by phase, right? So I've changed the coloring scheme. Yeah. So this is young, and this is old, and this is ribosomal protein. And what we get is a reaction coordinate. And we think this is the reaction coordinate for the evolution of protein folding. And what we can see in this result, I'm just going to jump to them, getting cut off here, is that protein folding occurred in a sea of RNA. Those secondary structural elements I talked to you about, I showed you, are enveloped in RNA. And we think RNA basically chaperoned protein folding, and that protein folding is an emergent property of interactions with RNA. And I didn't really show you this, but the RNA folding changes around basically these two polymers are changing together. The RNA is changing as protein folds, and as protein folds, RNA is, its folding is changing also. Not as dramatically, but there are definitely more sophisticated RNA folds around sophisticated protein folds. So that leads us to our sort of, what we believe is happening is that RNA and protein are what we call molecular symbionts, and that the evolution of protein folding was chaperoned by RNA, and that the evolution of RNA, I'm not saying folding because you had stem loops and things, but complex RNA assembly was chaperoned by protein. So that kind of leads us to how I've started to think about biology in general, This is because my friend Nick Hudd grows figs, and he gives me fig jam sometime. And so I started thinking about fig trees. And there's a symbiotic relationship between wasps and figs. The wasps pollinate the figs, and the figs feed the wasp embryos. And there's this beautiful, what's called a mutualism relationship. And this is all over biology, of course. In fact, you are involved in relationships like this with microbes in your gut. And there's something about mutualism relationships in biology that confers advantage. And we basically say that goes down to the molecular level, and that RNA and protein are molecular symbionts. All RNA is made by protein. All protein is made by RNA. And we could throw in membranes and other elements to this too, but that basically we define life as a molecular symbiosis. And that definition sort of comes from what we have learned from the ribosome. So with that, I think I'm terminated here, and I'll take questions. Thank you.
Okay, we have C, Andy here.
Thanks, Loren. That was great. I find myself wondering, as you describe the secretion through time, do we know much about the functional consequences of those modifications of the ribosome?
In eukaryotes, actually that's, some of it has to do, actually Charlie was just asking me, with ribosome biogenesis. I mean, people have cut these things off and say what happens, and ribosome biogenesis stalls out. But actually in our lab, we've taken those human expansion segments and we've done pull-down assays and say what proteins bind to them. And it's really interesting. I mean, it's like the proteasome is associated with the ribosome. It binds to that. And so we think they're kind of, especially those tentacles, and those are kind of docking sites, we think, for things auxiliary to translation. So protein quality control, things like that.
Hi, Loren. Yeah, nice talk. I'm concerned about one thing, and maybe you can help me out with this. When phylogenetic people are making sequences, some there are short branches and some there are long branches, but there's no such thing as a branch that has zero length. And it seems to me that if you're married to this accretion-only model, what you've done is put the E. coli ribosome, which as some at the bottom with zero length, and then accreted only. But it seems to me that when you're doing phylogenetic analysis, you have deletions and insertions, and I think they weight them pretty much equally. You have weighted only insertions and said there are no deletions. Is that right?
No, actually. I use that ES7, and really I should say this clearly, that the best estimate for the universal common core is the prokaryotic ribosome. And in some places you have things that are added on in bacteria, and in other places in archaea. And we can see that places there are deletions. I kind of didn't point it out because I was trying to.
Well, for example, you showed the comparison between the E. coli and the archaea, and I'm thinking, why do you assume that the E. coli is more basic and then you added something to the archaea? Maybe the E. coli lost something in the last 4 billion years.
Yes, okay, here's an example of exactly that. So this is in the small subunit, but you see this blue, E. coli has this, and sorry, this is Thermus thermophilus, and sorry. Anyway, yeah, these are both bacteria, so I can't really show that here. But yes, we have places where things get added on and taken off. I mean, here you can see a place where Thermus has lost, or the common ancestor, something got added onto E. coli but not Thermus, and then something got added onto Thermus but not E. coli. So, things are getting added on and taken off. And we have, no, you're right.
So E. coli is not at the bottom?
No, E. coli is not the bottom. I was just sort of speaking shorthand. It's our I was using it as an approximation for the bottom, but I could have used archaea for that, actually.
Yeah, very interesting. Do you think you can deduce which was the lifestyle of Luca, if thermophile, mesophile, psychrophile?
No, I don't think so. I mean, it might be possible once we know more, but what we know about Luca really what we know is the translation system, and you know, people who are trying to do, you know, does it live in a vent or whatever, like actually Sean talked about that paper. I mean, people are sometimes willing to kind of make assumptions about what Lucas is, but when I see that happening, it really seems to be model dependent. Like, you know, if somebody is a vent person, they're going to say Luca lived in a vent. I promise you this. And if they're, you know, I mean, that is just what is going to happen. And, you know, I just think you got to be very careful. And so that's why we use the universal gene set. I mean, that's data. We're not trying to make any assumptions about what was there. We're just saying these are the things. If you take ribosomal protein L3 from bacteria and you search in the strangest archaea you can find, you will find it, right? I mean, this is just, but people are very, you know, commonly trying to infer things about Luca. And when I look at the literature, it just seems like they always get the answer they want. So I'm very suspicious of those things.
Okay, so thanks for a great talk for molecular symbiosis. And I have a question for solubility of short peptide of ribosomal proteins. Okay, so before interaction in water, they have to be distributed in soluble form. So have you checked the short peptide or short peptide plus 2 beta strands matters solubility?
Okay, so we've made these peptides, and they're soluble. So we actually are studying the chaperoning of the peptide folding by RNA. The peptides are soluble.
Or if you can find such the sequence, you can estimate the solubility.
Yeah.
If most of them is aliphatic amino acid.
These are soluble peptides because, remember, they're living in a sea of RNA, and they're very highly charged. They have a lot of
RNA is soluble, but in the case of a peptide.
Oh, in general, a peptide? Yeah. Okay, so our model for the ribosome is the ribosome early on was doing non-specific. It was making, and I mean really non-specific. I mean esters. You can make polyester with the ribosome. It doesn't care. True. So I think the ribosome was making racemates of ester peptide. It was, and some small fraction of what it was making, and it could have been a very small fraction, because there wasn't a lot of competition in those days, right? We're talking about some small fraction stuck and conferred advantage, right? If most of it was insoluble or did nothing, it really didn't matter. If some small fraction stuck and conferred advantage and allowed the ribosome to fold better and function better and be more stable or whatever the selective pressure was, we don't really know, but then that would lock it in, and actually we believe that peptides got longer and the ribosomal RNA got bigger, and that this was a kind of a bootstrapping process that cycled in on each other. I didn't talk about the tunnel, but we can really see, we can infer sort of what was happening with the peptides by the length of the tunnel. And it's pretty clear that there was a sort of a cyclic process. Peptides got bigger, the ribosome got bigger. And it's just,
Great.
okay, I'm sorry, Batul, I'm too long-winded here, is that? Okay.
No. Two questions, Lauren. One is there any possibility, have you played with the idea that instead of the helix 25 being the ancestral state, that since bacteria are reduced in many ways, that entire archaeal bit is the primitive lucal for want of a better word, state, and that there was a loss of that upper part? And the other question is, have you thought at all about whether non-ribosomal protein synthesis could have been earlier or later?
Okay, first about, yeah, I mean, we sort of are using the assumption that things that are common are our best representation of the ancestor, but it's not necessarily true. So yes, you know, we really can't exclude that longer helix was ancestral. We can't. Okay, now what was the next question?
Is it possible that non-ribosomal protein synthesis preceded the ribosome so that you could have?
Well, I guess it depends on what you mean by non-ribosomal. I do believe that peptides were being synthesized abiotically before the ribosome. In fact, I think all the ribosome did was help what was going on there. And I think that possibly the original function of the ribosome was just to keep the two ends apart and stop the cyclization that kills your polymerization. So really all the ribosome, it looks like in the very early part of the ribosome, it was just a device where two amino acids could bind and they could cyclize, and the ends were not allowed to come together. So in that sense, yes, there was peptide synthesis going on before the ribosome.
Yeah,
but
I'm
you
talking
mean
about biological, you know,
I-
that still occurs.
I would have a hard time with that because that requires complex coded proteins, and I can't imagine where they came from. So, yeah, I'm not big on that model that there were intact functional proteins before the ribosome. I don't know. I think that's a difficult model.
Hey, Loren. Thanks for the nice talk. I was wondering just how you conceptualize the gradual buildup of the ribosome in general. There's these different kind of hierarchical levels that it seems like this progressive addition of subunits has occurred. Mm-hmm. So you showed the cool squiggly green peptides gradually accumulating secondary structure, and then it looks like there's some tertiary structure coming in. And so there's not any ribosomes today that only have the green bit. Is that right?
That's correct.
Okay.
Right.
And so as that happens, and in addition to that, at the same time, presumably, there's all these different ribosomal subunits that are being tacked on to the side. And so we see all these different subunits in archaea versus bacteria versus eukaryotes. So what's driving that, and what is the functional consequence of that, do you think?
Okay, well, what we think is happening is that you had this core and small things were added on. You know, and it kind of grew by the addition of small elements and small peptides, small pieces of RNA, and that it grew in that way. And we think there was a diverse population. In fact, and we're maybe not talking about the origins of the ribosome, we believe, could have been before RNA. The ancestor of the ribosome was not necessarily made of RNA. It could have been an ancestral molecule, and the product of the ribosome was not necessarily protein. In fact, we're pretty sure it was not protein. It was peptide. I mean, it was esters and things like that. And I'm not answering your question though. So tell me your question again.
It's just something that I wonder about when I look at all these different ribosomal subunits. You can draw a Venn diagram and find that there's all these different ones in archaea and bacteria and eukaryotes.
Okay, all right, ribosomal subunits but
but this is a different hierarchical level, but it's really in parallel to what you presented in that table of the green, yellow, and reddish.
Okay, but the
orange.
ribosomal subunits are the same in bacteria and archaea. That's where I'm getting confused by your question.
But some of them are different. Like bacteria have different ones than archaea ones, different from eukaryote ones.
Right? No, they don't.
Yeah.
The ribosome has accreted. Yeah, I'm not understanding your question. There's no different subunits in archaea and versus bacteria.
On the periphery there is. So you've got,
okay. Yes, there's expansion.
So you've got like how many ribosomal subunits?
Yeah. Okay. There's things added on. Yeah, we don't call those subunits, so I'm confused. Yeah. Okay. So there's expansion segments. That's what you're talking about that are added onto the surface of the ribosome.
We'll have to draw this out later.
All right.
But if there's time, I'll squeak in another question.
Okay.
So if I take that common gene set, and I throw that in a tube, what does it do, and what can that tell us about the first life?
If you take that common gene set, you can probably, and a little bit of other stuff you could do in vitro translation. So that tells you, I don't know, you can interpret that how you want, but I think you can't ignore it, right? If you have a model of the origin of life that does not account for the extreme conservation of the translation system and the fact that the LUCA had a translation system that is untouchable essentially by evolution, and if you want to take your next step back and say there was no translation system and there was a different biology, then that's what I find very difficult, because post-LUCA, the translation system is the most permanent system in the known universe, and if your evolutionary model goes a half a step backwards and abolishes it totally, I have a problem with that, as in the RNA world, for example, right? So the RNA world says there was no translation system, biology matured, and then a translation system was invented that suddenly became so important it's untouchable. And that just seems kind of incredible to me. I think this permanence and the resilience and the robustness of the translation system, you've got to take it back to the beginning. You've got to say that was built in. That's sort of, to me, that's the simplest model.
Well, I think we all agree that this was a fascinating talk. So let's thank Loren
Thank you, guys.
once again.